Taipale. I'm a professor at the University of Toronto and the Donnelly Center for molecular, cellular, and biomolecular research. Had my lab there for about 10 years, and I did my PhD in EMBL in 2001 to 2005 in Asifa Akhtar's lab.
So how did you get to know the EMBL and its PhD program?
I got to know it from my brother, who's a scientist too, and he started bugging me when I was an undergrad that I should apply to EMBL, and I was always laughing at him that there's no chance I'll do a PhD abroad, and I'm happy in Finland, and he kept bugging me, and usually you don't take any advice from your big brother. It's generally a bad idea for life, so you want to be independent and stubborn. So I didn't listen to him, and I actually started my PhD, and so I finished my master's in Finland, and then I started my PhD like directly after master's, because there it was very common that I would just do a master's then continue into a PhD, and then I kind of continued, and I was doing human genetics, and it didn't quite feel right to me. I was interested in human genetics, but it never felt like that exactly right fit for me. I was more interested in fundamental biology, fundamental sort of transcription, splicing, translation aspects.
So you decided to take the chance, and then you came to the EMBL.
I had already decided that I won't apply, but then for a postdoc I might then try to come to EMBL. But then I decided that no, I actually want to do it right away. So I decided that I will. Told my supervisor, Juha Kere, back then that I don't feel this is the right thing for me, and then I applied to EMBL, and he was very kind. He was kind enough to write me a very good reference letter, although I was his student, and then I got to the interview and got a position, and I have to say it was the best professional decision in my life.
See, so you should have listened to the advice of your
Well,
big
I told him
brother
that's the only time that advice from Big Brother was actually good. So yeah,
a key time. And what did you get involved in with Asifa?
So when I came for the interviews, I didn't know that Asifa was even because Asifa had just started or was just kind of accepted or started the job. So it was I didn't even know that she was at EMBL. So I was very interested in a couple of other labs, and I didn't know about Asifa, and then she came and gave a talk, like when the PIs gave talks to the students incoming or for the interview class, and I remember her giving a talk, and my passion had been at that point was really epigenetics and X chromosome inactivation, and that was actually something that I was really, really interested in, and I wrote like a seminar paper on X inactivation as an undergrad, and I was very aware of all the literature. So I was very excited there was somebody working on dosage compensation, not X inactivation, but like X chromosome upregulation in flies. So I was very excited about that, and then Asifa thought that I would be a good match, and she offered me a position.
So I was wondering, in reflecting of your time at the EMBL, what do you think was the factors that really contributed to the success of your PhD?
I don't think there's any key factor. It was just the environment generally. So first of all, the class that I had was full of really fun people who are fantastic scientists, and many of them have gone to do big things.
So then you moved to the Whitehead, and you, I guess, met a very different situation. What did you get involved in scientifically? Did you follow the path that you started in your PhD? Did you change completely field?
Well, almost completely. So
okay.
It was very much a change, because it was both sort of the lab-wise that when I started in EMBL, I was Asifa's first grad student. So it was just very small lab. We ordered the centrifuges and shakers and whatnot and installed them and all that. And then when I joined Sue Lindquist's lab at the Whitehead, I think there were 22 postdocs when I started.
So it was a large operation.
Yeah, it was a large operation and very well-oiled machine. So a totally different experience as a lab. It was a fantastic environment, but totally different from my grad school experience. But yeah, so I did change it because I worked on chromatin histone acetylation as a grad student, and then started working on chaperones, protein folding, protein homeostasis for a postdoc, so it was a different topic, and but first the methods kind of similar often and so on. So it was kind of I just thought that I didn't, I was really interested in epigenetics, but at that point I just wanted to learn something new. So
definitely. And I mean I'm not really in your field, but I got a sense from your research that you move towards more system approaches and more parallel approaches. And so what was your idea in moving there? Did you feel that there was sort of one gene or one process approach was too limited? What was the idea there to really massively move towards?
A little bit of it was serendipity. So one was that I had one project in Susan's lab with yeast genetics and yeast prions, and it got nowhere. And after 2 years, after 2 years being there, I really had nothing, more or less. And I was kind of looking for a side project, or at the same time was trying to develop something else, and then that turned into sort of a large scale approach. And also I think sort of the sort of systematic approaches fit better my interest and kind of my just personality, maybe, because I just like a lot of different things, and I could never imagine working on one thing only as forever. And I wasn't even sure if I wanted to be a PI as a grad student or a postdoc, because I was worried that I would have to work on like one thing or one pathway or one gene, and then kind of doing large-scale biology enabled me to kind of do a large screen and then figure out what comes out of it, and then just follow up on that, and it may be a different topic than what I imagined. And it kind of just one thing led to another, so kind of that way I became very interested in kind of large-scale approaches, and that's kind of what we do in my lab as well. And kind of sometimes I say it's also like my hypothesis-driven research may not be for me, because all my hypotheses have been wrong. So I kind of try to do sort of a hypothesis-free approaches, that I let the data tell the story, and then the data won't lie, and then I'll come up with a hypothesis later on. So
well, that certainly there's a big part of science that needs that. I mean, to build a hypothesis, as you said, you have to explore first. But the other thing that I think all these sort of massive parallel approaches are good for, I think, is also for, the translation of some of the observations, because I mean, you can of course use them as a tool. You can use them as a start for new drugs and other types of approaches that are translational. So I wanted to understand whether you are, considering also the translational part of science nowadays. I mean, whether you see some opportunities there. I mean, do you have some of the projects in your lab that are going towards that? Yeah. So definitely we think about it. Certainly, we're interested in kind of fundamental discovery first and foremost, but a lot of things that we do have applications, and it kind of started also serendipitously, and as a postdoc that I was really interested in just fundamental discovery, but then I found a way to kind of find drug targets or kinase inhibitor targets in human cells, and that became, I realized that it actually has sort of commercial potential, and we even tried to start a company back then, and it didn't work out, but I kind of saw a little bit of it, and kind of got very interested in that. So a lot of what we do is kind of related to that, and if we have potential to file patents or something, we do that, but of course we never let that kind of get in the way of science. But we have started one biotech company now in Toronto based on the research we do, and starting another one hopefully very soon. So kind of we do keep that in mind, and the results may be applicable, not the results or the technologies we develop kind of can be applied to drug discovery or as platforms for screening or so. So we're kind of interested in that as well. Okay, so what are you doing with these companies? I mean, what are you trying to discover?
So one is essentially it's called induction therapeutics, and the idea is that my lab works a lot on induced proximity. So essentially the idea that you could bring two proteins together with small molecules. Mm-hmm. And the best example right now is these PROTACs, or molecular glues that for targeted protein degradation, where you recruit an E3 ligase to a target and degrade it instead of inhibiting a target. And but we've developed these methods to kind of do that in a larger scale, and also kind of go beyond just protein degradation, kind of just thinking about the idea that what could you do with induced proximity if you could bring proteins together beyond just degradation. So activate proteins, rewire signaling, and so on. And the company is kind of trying to take that into kind of the next step with induced proximity.
I mean, I would think that already being able to trap a protein when it's a toxic protein or is a mutated protein would already be a good way to get rid of that protein. So, but is there other approaches? I mean, would relocalizing a protein, I don't know, cure a particular disease, maybe depending on mislocalization? Are there approaches of that?
Kind? So you can think about, for example, stabilizing proteins, mutant proteins, or rescuing their trafficking to the membrane, or taking really advantage of the whole breadth of biology. That there's so much happening. Biology is driven by these molecular interactions that change, and if you can modulate them, there's a lot of potential. And I think there's a lot of potential that we don't even know of. And a lot of these kind of most interesting discoveries in induced proximity have been these completely serendipitous findings that some molecule, instead of being an inhibitor, it actually brings proteins together. Like thalidomides in targeted protein degradation, that turned out that they actually recruit an E3 ligase to degrade proteins, or rapamycin that works by inducing an interaction between FKBP and mTOR, or there's some really wacko small molecules that have been discovered recently also that just bring together almost two completely random proteins but still have a biological effect. So I think there's a lot of discovery to be done there, and not only sort of rational approach, but also kind of just let the data give us some interesting insight.
I mean, it's interesting because there are two aspects that seem to be in what you're saying that seem to be almost polar opposite. On one side, the serendipity is, I think, the centerpiece of bottom-up research, right? I mean, I just look and I don't know where the discovery could come. On the other side, we have been discussing about, companies, startups, spin-off that really try to push research in a certain direction. So how do you see this dichotomy? I think this is an important discussion to be had, because we're always oscillating with people that try to say, well, we should only finance, direct research, top down, and I mean, fundamental biologists like me will always say, well, you have to give money to give also unbiased, serendipitous research, so that a new concept could emerge. I mean, what's your view on these things? You seem to be doing a little bit of both.
Yeah, I mean, there's definitely, interesting, there's some parallels, but definitely the approach is totally different, right? That in science, we're just standing in front of a vast ocean, right? We're on a beach in front of a vast ocean, and we try to discover something, right? And I'm very proud of being, going for fishing expeditions, right? That, people sometimes go, you get negative feedback that your proposal is, fishing expedition, but I'm card-carrying systems biologist, and I'm shameless in kind of doing that. That we'll go out and fish and see what we get out of it, and that's serendipity part of it, and part of this having good ideas where you might find something. And I think we need very, very strong fundamental research, of course, but on the other hand, then for the companies, it's totally different, right? So there you have, like, everybody has eyes on the ball, the same ball, which is to bring this molecule to the clinic, and then you toss vast resources into it and try to advance that as efficiently as possible. So it's a very different mindset. And for me, as an academic, I love the idea of just the wide open ocean, and I can go and discover something that may be totally unrelated what I do today, but that's where the data takes us. But certainly you can't do that in a company, because you have responsibility of the investors, and also your responsibility of bringing drugs to the market that will help people, that will be very different. So,
I mean, and what is your view on the interface between science and technology? Because I suspect that some of your approaches are also quite driven on technology.
Yeah.
I mean, I guess you need the latest technologies also for going for fishing expeditions,
Yeah.
like
Yeah.
in the ultimate proteomics and stuff like that. Is something you're developing in the lab, or you obtain that through collaborations? How do you deal with getting, the cutting edge technology for your science?
So we develop technologies, and sometimes we either apply or we develop new technologies depending on kind of where we are and whether we can answer the question with just standard tools. But I'm a huge believer in technology, that if you just think about the biggest discoveries in the last 20, 30 years, like almost every one of those is technology, going from RNA interference to iPS cells to CRISPR-Cas9 and iPS cells, right? That they're fundamental discoveries. How for example small RNAs and defense systems work, or CRISPR-Cas9 and pathogen or phage defense, or iPS cells. Just a fundamental idea of how you can turn any cell into a stem cell, which is fundamental discovery. But in the end, the bigger impact is actually the applications and what it enables, and I think technology really enables science. Of course, you need really smart people and really good ideas, but a lot of them are enabling in a way that when somebody discovers something, they don't even think about like how it could be applied all the way, because there's so many smart people that will then figure out other ways of using it. Mm-hmm. Like if you think about next-gen sequencing, that sequencing genomes is amazing, but if you look at what people use next-gen sequencing now, it's like all kinds of things that you just count molecules by sequencing DNA, rather than you're not interested in the sequence itself per se, but you use it as a counting tool or something like that. And there's so many clever ways of doing it, and people who developed it certainly didn't have everything, probably didn't have all of that in their mind.
The other thing that I was thinking about, I mean, you said it yourself. You had many interests. You have like a focus on unbiased approaches. The other thing that was quite fascinating is that you have toyed in your science both with a lot of molecular biology, nucleic acid biology, and then you have also done more recently a lot of work on proteins as well. And the interesting thing in reflecting of the history of biology is that we had an early phase in which it was all protein science, right? And then we have like the golden era of molecular biology, and now with a lot of the approaches we are moving back to a lot of protein science and possibilities with proteins. So I guess now you're also heavily working with proteins, as you said. So how do you see the development of this field, of engineering proteins, getting methods to actually analyze proteins? Where is the science and technology going?
Well, definitely. I would say that it's, maybe everybody says the same, but it's never been better to be a scientist than now. Like, I'm sure people said it 5 years ago, and I think we're going to say it in 5 years. But really, like, in the last 5 years, there's been amazing developments in, protein design and structure, AlphaFold and so on. And I think these are really, really enabling tools, right? And there's, I think future of proteins, design protein, proteomics and so on is very bright, right? The instruments are getting better and better, right? And I think there's a lot of cool things that we can do now that certainly wasn't possible before. And I can't wait to see what the future brings to us, kind of, and how the standards and the expectations change. That now we can do mass spectrometry with, soon in single cell level, and what are the expectations in 15 years from now to
I think a lot of these approaches, aside from the fact that they require a lot of resources, also they require more and more computation, more and more artificial intelligence, in a sense. So how do you see, first of all, are you already using artificial intelligence and heavy computing approaches to understand some of your data? And how do you see that evolving in your field, for instance?
So we don't use too much. I'm an avid, I use ChatGPT and Claude all the time myself, certainly, but we don't use too much yet. But something that I'm definitely interested in, especially protein design, is something that I'm very, very interested in. Actually, my current sabbatical has to do with protein design.
Okay.
But I think it's, I mean, there's no question about it that it will be even more important than it was before. Like, even for the last 20 years, computational biology or kind of understanding a large data set has been incredibly important, right, for any biologist. And you can never go wrong to, understand how to analyze data and how to, code or, write scripts and so on. But I think it's getting more and more important. But on the other hand, also, a lot of it probably will be commoditized in a way that protein design, and right now it's still, it can be done by the very top labs, and now, it's kind of trickling down to other people that, you don't need to be, David Baker from, to do this thing anymore. And maybe it's going to be so easy that, soon you can just say that design a binder for this protein and you click a button and you get it.
I see.
So it's commoditized in a way that then the only question is that what are you going to use it for? What is the application then, right? So when the barriers get lower, then of course your requirement for, hardcore protein design and understanding is much less as well. And so I think there's kind of two aspects of it.
That's an interesting thought, because, I mean, one would argue that some of the science become very difficult also because it's very expensive, right? You need the latest machines and, better approaches, and sometimes having a lot of money allows you to do things that you couldn't do in another lab. But you're making an interesting point. So, you're saying that, as you conquer that, then there will be essentially a sort of a scaling effect by which things are getting cheaper, and then more labs will be allowed to play with these approaches.
Yeah, absolutely. I think there definitely will be like much more accessible in the future. And I think it's really, really important also for sort of any new technology to make it as user-friendly as possible, right? Because in the end, the more adopters you have, the bigger impact you will have. And, the easier it is to use, the better, right? And that goes to any kind of a large-scale technologies or any kind of expensive technology or new technology.
Yeah. So what are you exploring in your sabbatical in terms of protein design? Are you looking at all the spaces to build molecules that the AI can explore?
No, I'm just want to think about like what could we use protein design for. Like I'm not necessarily for therapeutics, but just essentially biological discovery, or using them, for example, as functional proteomics screens, or using them for other kinds of things. And I'm kind of trying to think about it. We have a couple of projects that we have going on now on protein design, and kind of ask biological questions with designer proteins rather than using them for therapeutics or kind of regulating biology per se. So,
So you are exploring possibilities to enable the next process.
Yeah, exactly.
Projects.
Enable new projects.
And new science.
And really, like if we could design a binder to every protein in the proteome now, what would we do with it? Okay. And maybe that time will be soon. Maybe this. I don't know. Maybe it will not be possible for the next 10 years because we're not quite there. But maybe it's possible next year, and then we'd better be ready to do something with it, and have good ideas for it when it's possible. So I'm kind of these enabling technologies. The other one that I'm looking forward to is just having cheap DNA synthesis. That if we can synthesize, genome scale, we could synthesize DNA at the same cost as we sequence DNA. That would be completely transformative to a lot of things we do.
Absolutely. I mean, this will free up molecular biology in a sense. I mean, you can already do it with short sequences.
But if you could do that at the at 10 cents per kilobase or something scale, or like $100 for the genome or something, that would be really transformative for a lot of things we do. And I don't know when it's happening. I'm pretty sure that it will happen. But if it's next 5 years, if it's going to be next 25 years.
I mean, for one, you could play the little Craig Venter and build, your little organism. Yeah. I mean, because if you were at least get to the scale of a bacterial cell, you could just design your own genome they have done at that scale. But I agree. I mean, it would enable. Yeah. I mean, it will free us from the constraints of molecular biology.
Yeah, and students wouldn't need to grow any plasmids because you could just order the DNA and it would be in your bench next day. So forget about all the cloning.
Cool. So I think it's been a very interesting and enriching discussion. So maybe I'll ask you one last question, because we have this 50th anniversary, which is what EMBL means to you.
I think it means connecting people. That to me, by far the biggest impact has been just getting to know people. When I was here, I was only here for 4 years, but I've had lifelong friendships. Anywhere I go in the world for giving a talk, visiting a meeting, there's somebody I know, and I have lots of friends that I go somewhere, I stay with them, or I hang out with them, grab a beer, or talk about science. And just having the privilege of having friends all over the world is really amazing, and it goes far beyond science, right? It's a human connection as well, and I think that's really something that is, some places, scientists generally do that, right? Because you go for a postdoc somewhere and then move on. But I think EMBL, it's like yet one more level up, because just everybody comes from somewhere, stays here a little while, and then we go all our own ways then, but kind of have a little piece of EMBL in our hearts. So I think that kind of the human connection, I think, is even bigger than the kind of scientific connection, which is already very, very big.